chambers. Okay, various disease. So this is a movie for heart pump blood. Okay, various disease may affect ventricles and interfere with proper functioning. So if your heart is healthy, so this is the normal ECG you have in the hospital. But however, if you have ventricular tachycardia, so which means your heart beats very fast but regular, so your ECG may look like this. But if you have even worse called ventricular fibrillation, so which means your heart beats very rapid but chaotic, so your ECG may look like this. So both VT and VF are fetal. They can lead to sudden cardiac death. Okay. So every heartbeat is controlled by the propagation of active potentials. So this figure shows two active potentials generated by two consecutive stimuli. So every active potential has a steep depolarization, which is a rise of the transmembrane potential, followed by a shallow repolarization, which is decrease in membrane potential, right? So active potential propagation in the heart can be seen on two different scales, globally throughout the entire heart and locally between two adjacent cardiomyocytes. Okay, so this is a movie shows global active potential propagation in the heart, and on the right we can see two cells are communicated with each other by gap junctions, which is a low resistance channel connects intercellular space of two cells. Okay. So recent experimental study shows that if you remove gap junctions, and the mice can still live up to 2 to 3 months, and there is still signal propagation in the heart. People may ask, why is that, right? So there are many different hypotheses. One hypothesis is that if you remove gap junctions, you just remove one connection, for example, connectin 43, but you still have connectin 45 remains in the heart. So another hypothesis that, okay, something else may play a role. So if you remove gap junctions, okay, something else will replace gap junctions and force cell communication. So it's called ephaptic coupling. So what is ephaptic coupling? Unlike gap junctions is like channel, right? Ephaptic coupling is an electric field effect developed at the intercalated disc between two cells. Okay, so the left panel shows you have two cells. They are connected by the intercalated disc. The intercalated disc is very narrow and torturous, and there are different ionic channels lies on different pieces of the intercalated disc, right? So this intercalated disc can connect branched cardiac cells into a functional piece, functional one piece, like in the middle panel, right? But because the intercalated disc, we sometimes call it cleft. The cleft space is part of extracellular space because of the special geometry. So there is a large resistance between cleft and extracellular bulk. And then this resistance is reversely proportional to the cleft width. So if you have narrow cleft, you have stronger resistance and you have stronger ephaptic coupling. Okay, and then because you have a large resistance, it gives you a large voltage gradient between cleft and the extracellular space. On the other hand, right, you have a torturous geometry, so the ions may be depleted or they may be accumulated in the cleft space. So you have a large concentration gradient. So both of them together will give you a large electrochemical gradient between the cleft space and extracellular space. So then you have large electrodiffusion between the two. Okay, so ephaptic coupling, this terminology is borrowed from neuroscience. So around the 1970s, there is a debate in the neuron science asking about whether neuron communication is dominated by electrically or chemically. Right? So there is actually, I think so far, there is a direct experimental study shows that ephaptic coupling is real or exists in the neuron science. But however, in the heart, so far, I think people are working hard to show ephaptic coupling exists in the heart. But so far, I don't think there are direct experimental studies about this. And people, more and more people are interested in the physiological impact of ephaptic coupling in the heart. For example, people show that ephaptic coupling can increase conduction velocity and reduce conduction block when gap junctions are reduced. But however, there is some experimental study shows, okay, there are different localization of channels. For example, the fast sodium channel, RKI, and alpha calcium channels. They are important channels for X potential, so they are localized to the cleft. Okay, people may ask why is that? And they believe that these channels localization are directly related to the ephaptic coupling. Okay, but of course there are some gap in this ephaptic coupling study. For example, the impact of ephaptic coupling on arrhythmogenesis, which is something we care about, is not so clear. And the fundamental mechanism of ephaptic coupling is not so clear either. Okay, so this is our goal. First of all, we want to explore impact of ephaptic coupling on initiation, dynamics, and termination of reentry arrhythmia and the fundamental mechanism behind. Okay, so this is our model. We call it two-dimensional multi-scale electrodiffusion model with ephaptic coupling. So every cardiac cell is modeled as a cylinder. So they are connected by gap junctions to form a rectangular lattice. At each point, we define intracellular potential and extracellular potential. Meanwhile, we also record ionic concentration of sodium, potassium, calcium, and chloride for each point in the intracellular lattice and extracellular lattice. Okay, for the cleft space, which is denoted by cross, so they are the space between two cells in the horizontal direction, right? So, and then we call the extracellular cleft space, which is diamond, which is part of extracellular space, but stay adjacent to the cleft space. We call it extracellular cleft space. And the top panel of this figure shows lattice view of our model, and the bottom panel shows a circuit diagram for two cells. They are connected, of course, by gap junctions, and the cleft space lies in between cell IgA and the cell IgA+1. Okay, there is a large resistance between the cleft and extracellular space. So we have to keep a record, or we have to keep tracking of, for all the compartments here, we keep tracking of the voltage or called potentials, also the ionic concentrations of sodium, potassium, calcium, and chloride. Okay, so this is a multi-scale model both in time and space. Okay, so how can we model ephaptic coupling and electrodiffusion? So one of the major tools we use here is called Goldman-Hawkins equation. So it's called GH equation for short. And from the ionic current, you can see that this ionic current is regulated by both voltage gradient as well as concentration gradient. Okay, ephaptic coupling highly, as we discussed above, ephaptic coupling highly relies on cleft space between cells. Right? So, and then the cleft space communicate with extracellular space and adjacent cells independently. So we model ephaptic coupling by consider the diffusion current from the cleft and extracellular space, as well as the current across two adjacent end membranes. Okay, and then we also model electrodiffusion between intercellular spaces, between extracellular spaces, and between extracellular space and extracellular cleft space. The reason we do this is because in the diseased heart, the cell and its neighbors are very different in voltage and concentration. So that's why we model electrodiffusion between adjacent intracellular and extracellular spaces. Okay, so that's why we call it multi-domain electrodiffusion model. Okay, the equations are set out based on the current balance as well as conservation law. For example, right, for the intracellular space, right, we keep a record of the voltage and concentration. So for the voltage, right, we have balanced the current of this cell and its four neighbors using diffusion current. Also, we have to balance the current of the cell flowing across the end membrane, which is the membrane separating intracellular from the cleft, as well as the current flowing across the side membrane, which is the membrane separating intracellular from extracellular. So the end membrane and the side membrane, they function independently, right? For each membrane, we have to control the capacitive current and ionic current flowing across each. Okay, and then the concentration changes for each ion, right, is proportional to the flux flowing across intracellular spaces. Okay, for the extracellular space, we did something similar. And for the cleft space, right, cleft space is like, so this is cleft space. So this figure is not up to scale because cleft space is very narrow. And there is a large resistance between cleft and bulk. We have to balance the current flowing from the diffusive current from cleft to the bulk, as well as capacitive and ionic current flowing between these two, across these two neighboring end membranes. And the ionic concentration for the cleft is proportional to the flux across the cleft space. Very similar for the extracellular space. Okay, so for the membrane dynamics, we mentioned end membrane and side membrane. For the membrane dynamics, we use a more physiological detailed model. It's called the Laura-Rudy peak ventricular model 2007. We call it LRD 2007 for short. And in this figure, you can see there are many different ionic currents, pumps, and exchangers. And we also have a detailed calcium dynamics inside the cell. And then by experimental study, we see the localization of these three different channels. And then we also model the localization of the three different channels along the end membrane. And want to take a look at individual localization of each one and take a look at the impact. Okay, and then we start to go to the results. Okay, rain tree arrhythmia. So what is rain tree? Rain tree arrhythmia is a depolarization of waves traveled in the endless circle, right? The rain tree is fetal because the frequency of the rain tree is very high. It can even outpace the SA node pacemaker. So that's why the rain tree, if there is a rain tree happens, so the rain tree will dominate the heart and the heart will quiver. There is no blood pump out. So in other words, the rain tree is fetal, right? So this is a movie shows rain tree arrhythmia in experiments during optical mapping experiments. And so on the right is a simulated rain tree arrhythmia in the absence of ephaptic coupling. Okay, so then the question is, what does ephaptic coupling do, right? We see that ephaptic coupling can increase conduction velocity and reduce conduction bulk. So in other words, ephaptic coupling is beneficial, right? And then we want to see what happens for the rain tree arrhythmia. So back to 2022, we have a paper showing that ephaptic coupling can terminate rain tree arrhythmia in ischemia heart. So which means if you have less blood in the heart and you can have rain tree arrhythmia, but we want to explore why is that, or the mechanism behind rain tree arrhythmia and ephaptic coupling. So it's pretty hard to do that because in ischemia, many factors will play a role. So that's why we want to have a very simplified phenomena. Still have rain tree, but no ischemia, right? So this, and then we have generates arrhythmia or rain tree in absence of ephaptic coupling. And then we add ephaptic coupling afterwards and see what ephaptic coupling can do. Okay, so the first one is you have very narrow cleft, 5 nanometer, very narrow, and which means ephaptic coupling is very strong. And then you can see that the rain tree arrhythmia can be terminated very quickly. But however, if you're widening your cleft from 5 nanometer to 12 nanometer, so which means ephaptic coupling is weaker, and then we can see that there is a lot of self-collision. So you can see the wave collide with itself, right? After several cycles, and takes much, much longer than the first type, than self-attenuation, around 10 or 20 times longer. But still, you can see that ephaptic coupling can still terminate rain tree. Okay, so this is only 2 examples. And then we will ask, okay, if the phenomena we observe here is universal or consistent, so which means if you vary gap junction together with ephaptic coupling. Okay, and then we can see whether the termination pattern is the same or not. And then we can see this is a figure showing, okay, there are many different colors. The red color means, okay, the rain tree is terminated because of self-attenuation. And the blue means there's no termination. And, green means there is no termination. Blue means there is a termination because of self-collision. So from here, we can see the pattern. First of all, the pattern is pretty consistent. And then we can see that, okay, if you have very narrow cleft width or if ephaptic coupling is very strong, the range can be terminated because of self-attenuation. And then if you keep widening your cleft, something called green happens, so which means there is no termination. And if you keep increasing, so there is a termination happens because of self-collision. And if you widen even wider your cleft, or if ephaptic coupling is even weaker, there is no termination, right? Let me ask ourselves, okay, the pattern is consistent, but why is that? Okay, so first of all, I want to explore so-called mechanism. I think it's not the real mechanism, but it's like a physiology interpretation or explanation for range termination under ephaptic coupling. So from here, we can see that if you have very strong ephaptic coupling, and you can see the red curve. The red curve shows the AMP gate. The AMP gate is the activation gate of fast sodium current. So we can see that if you have very narrow cleft, the activation gates can be deactivated very quickly within 50 milliseconds. So which means, okay, if you have strong ephaptic coupling, it can deactivate the sodium current very quickly. So that's why range can be terminated. But if you keep widening your cleft, there are many different collisions, or the wave will collide with itself. And then, so the reason behind this one is a little bit tricky. And then we can see we pick up two different examples representing different gap junction coupling. And we keep record of the velocity in the transverse and longitudinal direction, as well as anisotropic ratio. Anisotropic ratio just want to show whether the propagation is more longitudinal preferred or transversely preferred. From here, we can see around 20 or 15 nanometer, you have high speed, and anisotropic ratio is still high. So which means these two factors play a role, which means the range is very fast. And on the other hand, the range is more longitudinal preferred. So that's why you can see a lot of collision with itself, and then the range can be terminated. Okay, and we want to see the impact of coupling on the initiation of range. Termination and initiation are two different biology phenomena. Termination means you have a range pattern first, and then we add ephaptic coupling later. But initiation is you have the ephaptic coupling at the very beginning, and you try to generate range using the same pacing protocol and the same initial conditions. We compare three different levels of ephaptic coupling, 8 nanometer, 20, and 150 nanometer. We can see that if your cleft is very narrow, ephaptic coupling is very strong, we can see the range cannot be generated, right? But if you widen your cleft, you have moderate ephaptic coupling, the range can be generated somehow, but it's not sustained. So it will dissipate after a while. But if you keep increasing your cleft width, so which means your ephaptic coupling is even weaker, and then we can see that you can generate a sustained and a single rotor, so the range is stable. So from here, we can see that strong ephaptic coupling can cause no range. Moderate ephaptic coupling give rise to non-sustained range. But weaker ephaptic coupling give you a sustained and a single rotor or single range. So this is when gap junction at 100%. Okay, if we reduce to 10% gap junctions, want to see what happens. So we still choose the same cleft width. So ephaptic coupling is the same level as before, but we can see that when D cleft equal to 8 nanometer, still strong, but you can generate range somehow, but it's not sustained. It will dissipate after a while. But in the middle, right, you see still 20 nanometer, but if you can see the range is there, it's sustained, and you can see multiple reentry, there is a very chaotic behavior like ventricular fibrillation, right? But if you have D cleft equal to 150 nanometer, even wider, and you just see a single and sustained reentry. Okay, so there are two different kinds of examples. And then we will take a look at the corresponding dynamics. So we use two-dimensional dominant frequency map, or called 2D DF maps. For the above two examples, we can see that, compare these two, you can see that D cleft equals 115, so ephaptic coupling is very weak. You have like more homogeneous DF map. But when D cleft equals 20, you have a fragmented DF. And the dominant frequency is reduced from 20 hertz to 10 hertz. And then you compare the bottom panel shows gap junction is 10%. And then we still observe something similar. Okay, weak ephaptic coupling give you a homogeneous 2D DF map. But however, in the middle, you have very fragmented DF maps. And if you narrow your cleft to 8 nanometers, still fragmented, but less fragmented compared with in the middle. Right? And from here, we can see that strong and moderate ephaptic coupling can give rise to fragmented DF maps. But the weak ephaptic coupling give you a homogeneous DF maps. Okay. And then we want to take a look at whether the phenomenon we observe is consistent. So we have different levels of gap junction coupling, 100%, 80, 50, 30, and 10%. We have different initial perturbations, so which means we have plus or minus 10 or plus or minus 20 of the initial perturbation. And then we also have different levels of ephaptic coupling. We can see that, okay, so when the cleft width is very narrow, you have either no reentry or non-sustained. You have only one chance to get sustained one. But if you keep widening your cleft, you have more and more chance to get sustained reentry. So the sustained reentry here may be single, maybe multiple reentry. We don't know yet. Okay, and then we see that, okay, we'll take a look at the corresponding DF maps. So in the DF maps, there are two measures. One is called a dominant maximal DF. The maximal DF and number of regions in the DF or fragmentation of DF maps are two important criteria for reentry dynamics. We can see that for different levels of gap junction, ephaptic coupling can reduce the maximal DF, even if you can see some perturbations, but the overall trend is maximal DF is reduced. And then we want to identify the number of regions in a DF map. And we keep a record of the cleft width corresponding to the maximal number of DF regions. We can see that the maximum number of DF regions happen in the middle. So which means when there is a moderate ephaptic coupling. So in the moderate ephaptic coupling, it can, of course, reduce the maximal DF, but also, on the other hand, it can induce instability and heterogeneity to cardiac dynamics. Okay, and then we may ask the reason, right? So from here, we plot refractory period and anisotropic ratio before reentry occurs. And we can see that for very strong ephaptic coupling, you can see the refractory period is enlarged. So with cause, there is no reentry. But if you reduce the cleft width, you will see that refractory periods is reduced somehow, but however, you still have very high anisotropic ratio, which means the conduction is more longitudinal preferred. So these two phenomena, the two factors will play a role that cause the reentry initiation is hard with ephaptic coupling. Okay, then we also take a look at the ionic modulation. The ionic concentration changes during cardiac conduction. So we can see that the blue lines denote extracellular ionic concentration, which means ionic concentration in the extracellular space. The red curves denote the cleft concentration. And we can see that the extracellular concentration is very stable, but right, there is a significant change in the cleft concentration. For example, the sodium concentration, there is a moderate decrease from 140 millimolar to around 120 millimolar. So, but you can see this panel, the panel B, shows that the calcium concentration in the cleft is almost depleted from 2 millimolar to 0. And the the potassium concentration in the cleft is largely accumulated from 2 to around 8 millimolar. So, and then from here we can see that different ionic concentration change in the cleft, and our hypothesis that the triggering mechanism of ephaptic coupling is depletion of calcium and or accumulation of potassium. Okay, let's do a quick summary. We show that ephaptic coupling is antiarrhythmic by terminate reentry, prevent the onset of reentry, and reduce maximal DF. But on the other hand, it can increase the transient instability and heterogeneity to cardiac dynamics. And triggering mechanism of ephaptic coupling is depletion of calcium and or accumulation of potassium. Okay, so right, as I explained, it's not a real mechanism. So this is a new project. So we collaborating with Professor R. Schultz at the University of Minnesota. We want to explore instability of wave trends and spare waves using spectrum theory. Okay, on the other hand, I want to take a look at the modulation of ionic concentration changes during arrhythmogenesis in the heart, as well as modeling the cleft or the intercalated disc geometry and see the impact of ephaptic coupling. Okay, I would like to thank all my collaborators. Alina and Stephen are two cardiologists working with me, and R. Schultz is a dynamical system person. Okay, our new collaborator. So I would like to thank my founding agency. So Daniel Cooney from UIUC is an assistant professor at UIUC, and we have been organizing a Midwest mathematical biology seminar. So if you have a student or postdoc want to give a talk there, or you want to volunteer to give a talk, feel free to contact us. So this seminar is held last fall. And every fall semester, we reserve some slots for the people who will be on the job market next year to practice their job talk and have some feedback from researchers. Okay, thank you very much. I'm available to any questions.
Okay. I think we have time for maybe one question whilst the next speaker sets up.
Thank you for the talk. I know very little about the heart, but if you, in a clinical setting, could suggest one of the variables like the gap junctions or the ambient ion concentration or the coupling, which do you think is the easiest to modulate in a clinical setting?
So in the diseased heart, the gap junctions are reduced in a heterogeneous manner. So for example, if you have ischemia in your heart, right? For the scar region, the gap junctions are reduced in a heterogeneous manner. The concentration changes and the ionic conductance are modulated, of course, as well. So that is known and observable, yes.